You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Dede Willis, CEO and board member, and Germany.Garner, colloquially known as Skip, the Chief Scientific Officer and board member of Orbit Genomics. And the website is orbitgenomics.com. So Dede and Skip, thank you for coming.
Thank you for having us.
It's a pleasure to be here.
Excellent. So what's the premise of Orbit Genomics?
Well, Orbit is really based on a series of scientific observations that I and my lab has made since the mid-90s when we started looking at the stranger parts of the human genome. At that time, I was co-directing one of the first human genome centers, and we had begun sequencing the human genome the hard way, and really noticed that there are particular pieces of the human genome which are very hard to analyze and to get right, and that are these things called microsatellites. Also, not only are they complex and difficult to measure, but they seem to be more sensitive and fast response elements inside of your genome, which then makes them very interesting and potentially very informative as markers for disease, health, aging, things like that. And so we've been pursuing that since then. And what came out of it is Orbit Genomics.
So the first premise is Orbit will sequence your DNA, and its uses are forensics, identification of possible diseases that you may be more susceptible to, or what's the focus of the analysis?
Our first, the product, as Skip mentioned, the technology can be used for quite a few different things. But our first go-to-market product is actually an aid to diagnosis for lung cancer. And we've chosen that because, one, there's a very tremendous need for the product. Currently, lung cancer is the world's deadliest cancer, and it's in part because there's no screening test for it. It's the only major cancer without a screening test. So it's typically diagnosed very late. And diagnosis today is done using a low-dose computed tomography test, a low-dose CT, but they have incredibly high false positive rates. So people generally who get a positive low-dose CT will need to get follow-on testing that's very expensive, and it's usually invasive. We've demonstrated in a pilot study that we can actually determine who is at very high risk for this so that we can confirm a positive low-dose CT. So that's actually our starting point, although we intend very quickly after that to launch tests that will give people risk of multiple cancers. Skip, if you have something to add, please do.
Yeah, the important thing here is that these million or so repetitive microsatellite regions in the genome seem to be very sensitive to the kind of state of your body, and it's environmental exposures and just how generally your all of your cellular systems are working. So as a consequence, it looks like they will be very informative and valuable as risk markers and as drug targets and other diagnostic tools for virtually every complex multigenic disease. And those are diseases that include cancer, heart disease, and a variety of neurological diseases. It's those diseases which being very complex, don't really have good high sensitivity, high specificity, high accuracy tests for them. And as a consequence, we think that exploiting microsatellites is a way to accomplish that for all of these diseases, which are the primary diseases that ultimately affect and shorten the life of almost all humans.
So what do you think is the reason that people have these microsatellite regions of their DNA? And if someone's predisposed to a condition, what does their microsatellite region look like as opposed to someone that's not?
Well, first, let me just define what a microsatellite looks like in the genome. So you know that there's 4 letters, C, G's, A's, and T's in the genome, and there's 3 billion of them. Well, occasionally in your genome, there are repetitive sequences like CAG, 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 and there are about 1 million of these spread throughout the genome. And these regions are under a fundamentally different biological process for maintaining them. So they can grow from 5 copies of CAG to 6, 7, 10, 20, or less. They also are known to cause a lot of diseases, Huntington's disease, Fragile X, a number of things. So they are directly causative of disease. And they are, as I said, because they're under different molecular and cellular processes than maintaining all of the rest of your genome, they seem to be very sensitive to the cellular state. And so it is that we have discovered and are exploiting to make new diagnostic tests, new companion diagnostics, and new drug targets.
So how in a microsatellite region, how many times is a particular sequence repeated on average?
Well, there are certain regions of your genome which have very long repetitive elements that can be thousands of letters long. We study those regions that typically have repeats that are 50, 60, 70 bases long, and therefore are easily measured by current next-gen sequencing technology, which makes discovery possible. And it's also the technology that we use for actually creating our clinical test. So these repetitive elements play a lot of different functions. You've heard of telomeres, which are the ends of the chromosomes, and there's repeats there. But we're really focusing on the repeats that are directly inside of genes in the part that actually codes for proteins, or parts that control the production of proteins or the amount that's there, and alters the structure of proteins. So they actually have a high impact in the genome when they vary.
Why would organisms have these repeats? What functions do you think they serve?
Well, first of all, it appears as though almost all organisms, except for the simplest viruses, have many, many repeats, these microsatellite regions in them. And I think that what they're there, this could be a philosophical debate about why nature put them there. But in reality, from a scientific standpoint, they appear to be there so they can, so that your genome can adjust on a very quick evolutionary timescale. In other words, for example, corn and plants like that have many more of these microsatellite repeats than humans do, and that's because they're out exposed to the environment. And should the environment change, become wetter or drier or colder, they have to respond very, very quickly. And it seems as though they use this rapid ability for these portions of the genome to expand and contract and enhance the survivability of a particular organism, corn, cows, or humans.
So if a sequence is repeated, 5 times, when it's used to create proteins, are you able to notice if the entire set of repeats is used or just one set of the repeats? For instance, could you selectively knock out some of the repeats? If a normal organism has 5 and you knock it out down to 3 or even 1, will that organism still function? How do you elucidate the mechanism of how these things are used?
Well, first of all, we study these where they've expanded or contracted, and in general, they're in places that are not lethal to the organism. So if it was, that cell would die, and we wouldn't be able to study it. But these expansions and contractions definitely alter a protein, which can alter whether it functions or not. And again, if it's an essential protein, it could be lethal. But otherwise, it could simply change how well that protein functions in your genome to do things like control growth or to control death, which are 2 hallmarks of cancer, or control how effective your immune system works, which is another hallmark of your ability or inability to attack a cancer that's in you. So these microsatellites appear to modulate the functions of many proteins as opposed to killing the function of any single protein.
Is there any correlation between the number of repeats and the modulation, or how it modulates protein?
Actually, there is, and we did studies a long time ago on dogs, and we found the gene that controls the length of a dog's nose, and that seems to be controlled by a microsatellite, where if you had X number of copies of this microsatellite, you have dogs with collie noses, and if you have Y number, you have dogs with pug noses. So in addition to controlling things like life or death of cells, they seem to play an essential role in many traits that govern us as humans. They could change things like the morphology, like in dogs that I mentioned, our ability to fight off disease, preferences, how well we smell, all kinds of things like that, So they seem to be a tuning knob that nature's put in there where we can control a lot of different functions and a lot of different traits and a lot of different diseases that play a role in humans and indeed all animals, virtually all life on the planet.
Have people tried to add additional repeats, to make like a Pinocchio dog, for instance?
Yeah, actually, we tried that. We wanted to make collie mice and pug-nosed mice, and it turns out microsatellites are, as I said, they're very difficult to analyze, and indeed, Orbit Genomics has, we spent decades developing algorithms so we can understand their function and measure them in the genome, but also doing things like using knock-ins, knockouts, or the new CRISPR technology. Microsatellites can be altered that way. We've been doing it in the lab and observing things like important disease or important genes that play a role in cancer X that seem to be have a microsatellite in them that we find is important for that cancer aggressiveness or growth rate or something like that. As we go in with CRISPR and we change it from being an abnormal number of repetitive elements to a more conventional number, then the growth rate of those cells slow down. And if we alter it, the growth rate of the cells speed up. Again, a very much a hallmark and an indication of cancer.
Have you seen that, for instance, a non-cancerous cell may not have a microsatellite, but a cancerous cell will have a microsatellite? Or is it just the number of repeats, the size of a microsatellite that modulates the aggressiveness of cancer, for instance?
It's the latter. It's not that a microsatellite tends to disappear or not exist. It's that it expands or contracts and then alters some protein or some regulatory element sufficient to push a cell from being normal into a cancer state.
What about the transcription of a given microsatellite? Let's say there's 5 repeats. Will the transcription always be all 5 repeats? Will the transcription sometimes include part of a sequence that's not part of the microsatellite? the microsatellite plus, an extra sequence on one side of it or another? Have you seen if it's very modular, how they're used?
Yeah. So first of all, there are microsatellites that are in the coding regions of genes, of proteins. And then there are ones that are in the intergenic regions or intronic regions that are kind of outside of the sequences that are transcribed. But there are thousands of microsatellite or repetitive elements that are in the transcribed regions of genes. And indeed, they're flanked on either side by non-repetitive sequence. And when that particular sequence is being transcribed, it just goes into a repetitive element, and then the amino acid tends to be repeated. So because most repeat elements that are within the coding parts or the transcribed parts of genes have the repeat element is either 3 or 6 or 9, and so thus the amino acid sequence can be repeated many, many times. Now, in particular, we've made certain discoveries where the microsatellite in a coding region will expand not by 3 bases or 6 bases, and therefore keep reliably making all the amino acids afterwards, but it's something called a frame shifter, where if it adds or deletes only 1 base out of its entire repeat, it will cause you to shift into a frame, and then after that, all the amino acids are really nonsensical, and so it really disrupts the function of that protein. And so those are the ones that are the most impactful, and in particular, those are the ones that we study for things like drug targets. But many of the other ones that don't shift frames or are in regions outside of that's transcribed are nonetheless very informative and play an important control function in your genome.
Have you seen microsatellites that repeat in another region, a separate, disparate region? We've got 5 repeats here, and then 1,000 base pairs away, we've got another microsatellite that's exactly the same sequence.
Well, we have not seen where there is a linkage or coupling between one given microsatellite and another. The way we evaluate these things tends to tell us the individual value of any particular repetitive element. Now, as you know, a long time ago, in the early days of really genetics and exploiting DNA, microsatellites were used as linkage markers in order to be able to identify variants and changes in your genome that are causative of disease. Now that in this era where it's easy to sequence your genome and figure out what do the variants do, we really don't use linkage analysis that much, but nonetheless, we still look at correlations between individual variations at microsatellites, for example, or SNPs or other things, and a particular trait like having cancer or not having cancer.
Have you tried to maybe insert a non-coding part of the sequence right in the middle of a given microsatellite to see if that disrupts it or if that alters it?
Well, we haven't had to do a lot of those studies. A lot of those things have actually happened in nature. I mentioned Fragile X. And there are a number of other diseases where it's a sensitive area, a break point where you can have in this repetitive element, have it break and then recombine with another portion of another chromosome. And those things can result in rather, some very severe diseases. And so they are susceptible to rearrangements in your genome. There's hotspots for places where your genome can rearrange.
Have you tried to deliberately add a duplicate microsatellite in a different region of the genome to see what that effect would be?
No, we haven't. It sounds like a cool idea. I know that people have done things like put in duplicate genes, and a lot of genes have microsatellites in them. And so, whether it's a dosage effect because you have the whole gene there or not, certainly the microsatellites continue to play a role in whether you have one or multiple copies of a given gene.
Well, why do you think microsatellites can modulate things like the length of a dog's nose? Do you think that, if you have a certain sequence that codes for a given protein, because it's repeated more, that means just maybe 3 times that amount of protein will be transcribed and created and raised that level, and that modulates, the morphology of a dog's nose or other things, or the expression of something, just because there's a lot more of it?
You actually got it perfectly. Whether you're modulating the amount of protein or the effect of a given protein because you've altered its sequence slightly, if you look at SNPs or single base variations in the genome, largely they're things that either affect something or does not affect it. It either has a C there or a T there. However, the microsatellites have a continuum of possibilities that you could have 5 copies, 6 copies, 7 copies, 10, 20 copies, and that allows you to have an adjustment or a tuning knob for, again, the amount of protein that's made or the structure of a protein that ultimately can alter things like the morphology of a dog nose or your susceptibility to disease or whether or not a cell really becomes cancerous or not. So indeed, it's really probably nature's fine-tuning knob for a lot of things that go on.
How does this interact with epigenetic changes, methylation, histone deacetylation? Does it happen differently for these microsatellites and the phenotypic expression of them?
There have been a lot of studies, especially about 10 or so years ago, looking at methylation, especially at things called CPG islands, which tend to also have a lot of repeats in them, like C's and G's, and so there seems to be a correlation or some coupling between certain epigenetic processes and the fact that they take place at microsatellites. So microsatellites not only can change things because of the fact that they can alter their genomic sequence, but they also may alter their various epigenetic processes that can alter, their methylation status or something like that, and play a role, which of course is can be turned on or turned off in any given cell at any given time.
I mean, do these microsatellites lend themselves to epigenetic modulation, or do they dampen its effect? Any interaction there?
Well, I don't think that's known right now. I do think that people know there's an association, and indeed, one of the reasons why I started really studying microsatellites was I was very disappointed in the big discrepancy between the known amount of genetic components of a disease, which is measured by family studies, whether things are passed down or not, and whether there are genetic components, whether it's a single base variation or some epigenetic process or something else that could explain or account for that amount of that known or suspected amount of genetic component just can't be explained. And so it was for that reason I really turned my attention to studying microsatellites, because there was many of the genetic components of diseases, especially these complex diseases, are really unexplained to date.
Have you seen a correlation in how easily a particular pathway is upregulated or downregulated, if it's associated with a microsatellite or not?
Yeah, we've actually, again, I said there's microsatellites are in coding regions, but they're also in regulatory regions. And we study that as well. And we find very frequently microsatellites that are actionable or informative, that really seem to play very much a role in disease, are either closely associated with a regulatory element or some other functional element in your genome. And indeed, we've actually published on a microsatellite that is in the regulatory region right next to the coding region in certain genes that that control susceptibility to breast cancer. And so in that particular case, the expansion or contraction really alters a transcription factor binding site. And so it works by the fact that the expansion or contraction makes it so that various other proteins can or can't bind as well to particular genetic regions. So there's all different ways in which these microsatellites can act, or various mechanisms through which they can act to cause disease, to affect your response to disease, and to change things like traits and a variety of other things like that in your genome.
As if you look at a family, do you see changes in the microsatellite length, throughout various generations? Have there been any studies done that have looked at that?
Yeah. So actually, it was an experiment I did a number of years ago, right after the introduction of next-gen sequencing, that where I made a critical discovery that is really the basis for Orbit Genomics. And that is, we were studying mother, father, daughter, families like that had been sequenced. And of course, we know that Mendelian inheritance means that you get one copy of your genome from your mother, one from your father. So therefore, a daughter should have that at every place in the genome. But what we discovered was that using the latest next-gen sequence data, that indeed the sequence that was received by the daughter appeared to not agree with the parent's sequence 80% of the time. In other words, it was correct. In other words, Mendelian inheritance only worked 20% of the time for microsatellites. Well, it turns out that's not a function of nature. That's a technological flaw that we discovered and then discovered a workaround. In other words, the algorithms that are used to analyze and assemble the raw DNA sequence from NextGen gets it right only about 20% of the time. What we did was completely change the way in which we assemble these genomic reads from NextGen sequence at microsatellites, which drove our accuracy from 20% to 95% plus. What that means is that for the first time using NextGen sequence, we could actually make discoveries. Because if you have 80% noise, you are not likely to see any correlation or understand causation or anything else. So once we established these new algorithms to correct the human genome sequence, that opened the floodgates for many, many discoveries that are the basis of the new diagnostic tests for lung cancer, for other cancers that are part of Orbit Genomics today, that are part of a pan-cancer for all cancer risk diagnostic for Orbit Genomics, which is tomorrow, and also the basis of new drug targets and companion diagnostics for various drugs and their effectiveness, which is the day after tomorrow for Orbit Genomics.
Yeah, that's amazing. That's a huge step forward. I don't know if it's worth chasing the last 5%, but where do you think that that error comes from?
Well, the question is whether it's an error at all. So you know that you're supposed to really inherit one copy from your mom and one copy from your dad, but there's also a spontaneous changes that go about in your genome. And so that last 5%, there may be a few percent of that which is technological, which we might improve in the future, or they might simply be the fact that that there are these spontaneous variations that happen in these sensitive regions when, of course, you are making a new human that indeed might be real. Now, the reason why we think that that's probably happening, that there's these spontaneous variations, is because we have studied the evolution of an individual's human genome over the course of their life. We published a paper on genomic aging, where we actually looked at and we did next-gen sequencing of various individuals, at one point in their life, 20 years later, 40 years later. And we identified that, of course, you accumulate a lot more errors as you age. So things like SNPs you see, but microsatellites are particularly susceptible to changes as you age. And so that became a kind of an interesting area where perhaps, and it looks like we can very much exploit that to measure an individual's genomic age and how fast your genome is aging, and whether or not you can change how much your genome is aging. Again, the way to do that is if you have have a healthy lifestyle and all the things are working correctly in your body, you're likely to make fewer mistakes and errors as your genome ages. However, if not, then you can make more mistakes. And of course, some of these mistakes will turn into things like cancers or other neurological diseases or heart disease or something like that. And so another application of microsatellites is to measure your genomic age, your aging rate, and how you might change your aging rate by things that you could do in altering your lifestyle and thus altering your longevity. So these may be an important readout for longevity and for how you might adjust what you're doing or what your doctor's doing to maximize your longevity.
That's really interesting. So what did you notice that certain regions of a person's genome did not change as they age? Did all regions change, and some to greater or lesser degree?
Well, I think it's too early to know that without a much bigger study that we'd have to conduct. But there are certainly regions that are very protected, that really, if you altered them, then it could be lethal. And so therefore, you wouldn't see that in a study because the person would have died or the cell would have died. But indeed, I think that a thorough study of this will allow Orbit Genomics to identify the most informative microsatellites throughout your human genome that are most susceptible to change due to things like lifestyle or proper medical treatment, etc., that could alter and affect your overall aging, your longevity, your quality of life, your health.
Do you see any potential for reversing some of the accumulated alterations to the DNA as someone ages?
Well, the answer is probably. We have seen, and in this aging study, where we can alter the rate of changes one way or another. But certainly, again, microsatellites are quick response elements that can respond faster than any other parts of the genome and expand or contract. And so that means that they, because they're fast response elements, they're very sensitive to the overall conditions of a cell. They very well may be able to be to expand and contract and be a direct readout of things like return to a healthy state or diverging from a healthy state. Right now, we're able to measure the genomic aging rate, and so that's an indicative measure of how well you're doing on things like lifestyle and maintaining your body. There may be additional layers that we can capitalize on to refine those as measures of your aging rate and your longevity potential.
What sets the aging rate? What factors do you think are the biggest levers on the genomic aging rate?
Well, I think some of it has to do with a combination of the status of thousands of genes and genetic variants that you inherit, but others, of course, have to do with environment. Now, we know that there are certain diseases that cause increased aging and cause these people to not only age faster, but they're susceptible to diseases of the aged. And one of those is something called Fanconi anemia, which we published a paper on a few years ago, where Fanconi anemia in particular is a defect in some genes that control how you repair changes in your genome, especially repair uncontrolled expansions or contractions at microsatellites. And so that particular disease is illustrative of what can happen when you really break part of your error correction mechanism, resulting in very rapid aging and susceptibility to diseases as a child that are normally reserved for people who are, seniors. And so that is an extreme example, but for the rest of us, there are thousands of things in your genome that work together to control how many errors are made and how well they're corrected when cells are replicated. And of course, that's not only the genetic component in there, but there's also this environmental component that if you're stressing cells when they're replicating, then they tend to not replicate perfectly, or they tend to not correct changes that are made perfectly. And so things like cellular stresses, and cellular stresses are environmental. Everything from what we eat to what we're exposed to, carcinogens, mutagens, good wine or bad wine.
But have you sat in front of a printout of someone's genome now and then 20 years ago, and maybe color coded the differences and looked at it and saw, what's different about it? Has anyone taken that approach?
Well, that's exactly what we did when we did our aging study and published a paper on that. And to kind of put it in some quantitative numbers, that it appears as though, several people in the study were accumulating new errors in their genome, especially at microsatellite regions, at a rate of about 1,000 per year. Okay, now you might think that's a lot, but that's 1,000 changes out of 3 billion letters, and so only a small fraction of that 1,000, may end up in very important areas. But nonetheless, it only takes a few to really alter your life. Now, we did find one individual who was genomically aging at 10 times that rate. So there was either something that this person was being exposed to, or the fact that his germline genome, the genome he was born with, just didn't perform as well in making certain that cells replicate their DNA or repair their DNA perfectly.
Has there been any call to sequence someone every year, starting at right when they're born? That would be fascinating to see maybe a rise and fall of errors, or just a slow continual accumulation, or, a punctuated slow, fast accumulation of errors.
Well, that's exactly what the future of Orbit Genomics wants to do for everybody. We know that you get a lot of the story about your susceptibility to disease and your response to drugs and other things through the germline that you're born with. However, because you're aging continuously, then it means that you really should be making that measurement occasionally. And I agree with you. I think that in the future, all of us will not only have our genome done, but we'll have it done every year, so that we can not only measure our genomic aging rate to identify perhaps things that we can do to make us age genomically slower and therefore have less errors that could lead to future disease, but we could identify certain variations that would suddenly trigger a very high susceptibility to a particular disease or cause a disease. For example, the creation of a new cancer cell, because your genome has now taken on some new variations that are variants that really make a cell more cancer-like, whether it changes growth rate to be higher or lower or evade your immune system. But indeed, I think the future of precision medicine is to measure your genome very frequently, a year or so, and in particular focus on accurately measuring in your genome those regions that are most likely to be susceptible to changes and most likely to be very actionable, that can alter your susceptibility to disease, and those are likely to be microsatellites. So I think the future is sequence your genome many times, but also focusing on getting the microsatellites very accurately when you do that.
Yeah.
Are you doing that?
I
mean, you have the ability to do that like all the time. I would think that you guys, maybe even secretly, but you'd be doing it all the time in yourself just to see what's changing.
Well, the truth is that that one of the things that Orbit has accomplished is that once we've done the discovery process and found certain microsatellites which are really diagnostic for your susceptibility to a given cancer, lung cancer, for example, when we do that, we use publicly available data, which tends to be about, technically, when you do a next-gen sequence, you get about 30 copies of your genome to put it together. And so that's pretty good. But when Orbit Genomics takes those things that we've discovered from the publicly available data and use those, put those into our assay, when we next-gen sequence those particular microsatellites for our patients, we sequence those at over 500x depth. So there's unprecedented accuracy in our particular test, which then gives us very, very high, accuracy, sensitivity, specificity, and ability to really inform clinical decisions and personal decisions.
And if I could just interject, one of the studies that Skip did and published on, we actually measured individuals and over a course of time, and one particular individual, we don't know what he was doing at the time, so we can't correlate it to any specific actions, but he started off with a relatively healthy genome, and then 7 to 10 years later, he was extremely unhealthy or much less healthy, had a lot more mutations, a lot more aging going on. But then another 7 to 10 years later, when the genome was resequenced and reanalyzed, he was actually back to a better state than he was in the first sequencing. So we know that people's genomes do change, and what you're suggesting is exactly what we would like to do, is to be able to go out and get regular testing to see what is, you're born with some risk, but then you're developing it through your lifestyle as well. And are there things you can do to change? And is your disease, if you know you're elevated risk for something like colon cancer, what if you change your diet, which is what everyone recommends? But today, if you change your diet, you have no way to know whether that's effective other than just waiting to see if you'll ever get colon cancer. So it's those types of things that is in Orbit's future.
Well, I was going to joke and say that you should get sequenced before and after your in-laws come to stay with you for a month, and see the breakdown.
Well, I understand that. That's a considerable stressor.
Well, you did bring up something really important, though. So you're saying you do have at least one person where their state regressed or improved, because that's really interesting. I would think that we would only experience either epigenetic changes that don't affect the underlying DNA or negative changes, like a degradation, like you said. But are you saying you actually saw an improvement in someone's underlying DNA, and it must have been likely because of, whatever their environment? Is that what you're saying? You've seen actually an improvement, a reversal of damage?
Well,
yeah.
The answer is yes. And again, the reason why is that changes in your genome can come about because when it's replicated, it's not replicated correctly. But also, there's lots and lots of genes and processes that continuously monitor the state of your genome and go in and correct certain changes. And sometimes they correct them very well, sometimes not. And so it's entirely possible that changes can be reversible because of things like error correction processes managed to work better, and return them to a more normal state.
That makes sense. And then, sorry to hit you with 80 million questions. It's just so interesting to me. When you sample someone to, look at their DNA, are you looking at their blood? And have people tried looking at blood and skin and maybe, cheek swab and comparing them, each time someone's sampled? Because, your blood cells, for instance, if you're sequencing them, they may be changing in a totally different way from your cheek cells or your pancreas cells or your eye cells.
You're absolutely right. So for Orbit Genomics, the best and easiest and quickest, most informative test is to look at your blood, because your blood really samples a lot of your body as well, and it's also fairly non-invasive. And of course, you can get your genome out of a cheek swab too. And so we've done both, and reliably gotten good sequence out of both. But also, we have studied, indeed, the aging study that I mentioned. A lot of those samples came from layers, some tissue below the skin. So therefore, part of the things that may be going on there is environmental exposure. Now, there are certain parts of your body which are really exposed to the environment: your skin, certainly your lungs, and certainly, your gut. So those are the things where, you maybe want to stay out of the sun, or you shouldn't smoke, or you should eat a right diet. All those things can alter, especially the tissues that are very exposed to the environment. But we have looked at and sequenced and looked at the germ lines and things like tumors across all the different cell types in the body and all the different cancers. And indeed, we find some microsatellites that are informative and unique to a given cancer. But time and time again, we're finding certain microsatellites that turn up in multiple different cancers for being risk factors. So there are, without a doubt, certain repetitive elements, certain loci, certain microsatellites in your genome that are universal markers for your propensity for getting cancer or getting other complex multigenic diseases.
Have you compared at the same time, again, skin, cheek swab, blood, tears, I don't know, multiple places on the same person, and looked at to see if the damage profile was different in these different types of cells, the microsatellites, the proliferation or number of them or character of them was different?
The answer is yes. And we've put some little bits and pieces in papers in the past, but hold on to your hat. Very shortly, we are going to be submitting a paper where we actually sampled tissues across multiple places in your body. And we actually did that on cadavers in our anatomy lab at the medical school. So, and we produce, in that particular case, it's really to help our doctors be prepared for the future of precision medicine, where they have to really understand and interpret genomic data, especially things like microsatellite-based tests, right? So we have a paper that is going to be submitted soon, where we tested, again, tissues from all of your body. And the bottom line on that is, of course, your genome is your genome, and it's very similar across all those tissues, but also from one tissue to another, there could be thousands of differences in your genome between one and another. Again, because they're all under different environmental conditions. They expand out of different earlier populations of cells and things like that. So in general, all the cells that you have are more or less genetically the same, but there are a lot, hundreds, thousands of differences, and those can account for perhaps your susceptibility to why certain tissues are more likely to get cancers than others.
Well, very good. This has been a great call. I didn't hear from you guys once. That's proprietary. We can't tell you. And you guys said yes to everything. It sounds like you're experimenting and trying a lot of different things. And
I mean, the work that you're doing to help people with their cancer is also honorable. So I appreciate you guys coming. What's the best way for people to find out more and get in contact?
Through our website, which is www.orbitgenomics.com, or our email is info@orbitgenomics.com.
Okay. Well, Dede and Skip, thank you so much for coming. It's been a great call. I appreciate it.
Thank you.
It's been a pleasure telling you our story, and I love to talk about science.
I noticed. Me too.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.